Welcome back, everyone. So we are continuing on Chapter 6, Reproduction at the Cellular Level. So we're going to finish, talking about cellular division with prokaryotic cellular division 6.4. So looking at how our bacteria and archaeans divide their cells. So this will be a little bit different because prokaryotic cell division is a lot less complicated than eukaryotic cell division. I mean, it starts the same, where the prokaryotes need to replicate their DNA, and then each copy is allocated to the daughter cell, just like in eukaryotic cell division. But there's no mitosis that takes place because we don't have a nucleus or anything involved. So it's just basically the cytoplasmic contents are divided evenly into the new cells. So it's a little bit different because there's no linear chromosomes. It's just singular circular DNA, and there's no nucleus involved. So we don't have mitosis. Instead, we have a process called binary fission. And then bacterial cytokinesis is a little different because we have a ring which is composed of something called FtsZ. So we'll talk about that special protein and how there is kind of an ancestry involved with it, with our own microtubules and mitotic spindle. So by the end of this video, you should be able to describe the process of binary fission in prokaryotes, as well as explain how this FtsZ protein and tubulin proteins are examples of homology. So let's start out by just kind of going with an overview of prokaryotic cell division. So just like in eukaryotic cells, prokaryote cells, they need to divide themselves, right? So it's basically for us humans and animals, we're multicellular. So we are doing cell division in order to grow and to replace damaged cells. But for prokaryotes, they are undergoing asexual reproduction. So they're completely dividing themselves to make another one of themselves. Okay, so this is a form of asexual reproduction. And so they're propagating by a form of what's called binary fission. And so this is a form of asexual reproduction. And it's a simple process. It's just basically they're taking their genomes, their complete set of DNA, and they're replicating it. But remember, their DNA is circular. It's not linear like our DNA. So they have, their circular chromosomes, and so it needs to be replicated first. And then once their replicated DNA is present, the cell will get bigger, just kind of like ours do. And then the contents of the cytoplasm are divided. Okay, so you can see that the process is, pretty simple. Okay, we don't have a nucleus involved. We don't have the linear DNA. We just have circular DNA. So no mitosis is taking place. All right. And in this case, you can see that we're creating two individual bacterial cells in this case because it's a prokaryote. So this is a form of asexual reproduction. So in binary fission, it's a lot less complicated. We don't have, the prophase, prometaphase, metaphase, anaphase, telophase stages like we did in mitosis. There is no mitosis, so it's a lot less complicated and it's a lot quicker. So our bacterial cells, they can replicate themselves very, very fast. So they can go from one cell to, a whole colony of cells very, very quickly. So think about, when you have a bacterial infection, it can, you can get exposed to, for example, streptococcus, which is, strep throat. You can be exposed to it, and then just a couple days later, you end up having that sore throat because you have the colonies of bacteria that have developed and are starting to wreak havoc with your immune system. So, they divide and divide and divide really, really quickly, and now you have billions of these little bacterial cells colonizing, your respiratory tract. So this can happen very, very quickly. So what happens through this process? So let's take a look at our figure over here on the right. And, this figure is from your textbook, by the way. So, if you're doing this on like a cell phone or something, you can't see very clearly, this is a time when you can get out your textbook. On a laptop or something like that. It's better to see it. Okay, so here we have a simple prokaryotic cell. So remember, prokaryotic cells, they lack a nucleus. So where's their DNA? It's just in the nucleoid region of the cell. And remember, our typical prokaryotic cell just has basically 4 components. It has the cell membrane, the cytoplasm, the DNA, in this case a circular loop of DNA, and then the proteins. So we can see here we have our cell membrane, we have the cytoplasm, we have the DNA in the circular loops, and then we have the proteins. So in this case we have DNA replication taking place. So the replication, since it's circular, it's going to be taking place starting with the origin of replication. So the origin of replication is going to start out with a little bubble, and then it's just going to start spreading from there. So, we start from either side, and it just spreads until the entire loop is replicated, and we end up with 2 loops. Okay. So it continues in either direction, both directions, until we have a complete duplicate of the chromosome. Okay, so now we have 2 loops. Also, you'll notice inside of our prokaryotic cell, we have this special protein called FtsZ. So FtsZ, it stands for filamentine temperature sensitive mutant Z, which we'll talk about in the next slide, but it's just a special protein that we'll talk about in this sequence here. Okay, so pay attention to what's happening with these proteins. Okay, so we can see that the DNA in the second step has fully duplicated. Okay, also notice that these FtsZ proteins, what are they doing? They're migrating towards the loops. Okay, so they're migrating towards the midpoint of the cell, towards the loops. Also, you'll notice that the cell is elongating. Okay, so that's basically the second step that's happening. All right. The third step is, you can see that the loops are separating. So they're moving to opposite ends of the cell. The FtsZ proteins, they're starting to form a ring in the middle of the cell. Okay. And you start seeing this cleavage furrow or pinching of the cell take place. Okay. And then finally, the FtsZ ring directs the formation of what's called a septum. So the septum continues to divide the cell. So that cleavage furrow is forming from the FtsZ proteins. The plasma membrane and the cell wall starts to accumulate, and you have cytokinesis, the complete separation. Okay, so the septum is complete. The cell pinches into 2, forming 2 daughter cells. All right. And you can see that now the 2 daughter cells, they look pretty much like the original cell. So you have the identical DNA inside. You have those FtsZ proteins in there so that they can divide again. They're identical to each other as well as the parent cell. So this is identical copies. And that's it. very simple. Okay, so there's no, mitotic spindle involved. There's no, dissolving of nuclear envelopes or anything like that. There's no stages. So you just have basically the FtsZ proteins moving towards the middle, forming a ring. The ring is helping form that septum, separating the two cells from each other through cytokinesis. That's it. Pretty simple. All right. So that is binary fission in prokaryotes. Now, we're going to talk a little bit more about these FtsZ proteins because they serve a bit of like a evolutionary role in our microtubules or tubulin proteins in our eukaryotic cells. So what is FtsZ? So FtsZ is a tubulin-like protein component, and it plays a role in the prokaryotic cytoskeleton. So it's important in cytokinesis in prokaryotes, right? So we talked about how they form the FtsZ ring, which helps start cytokinesis in binary fission. And again, here's the full name. So the abbreviation FtsZ stands for filamenting temperature sensitive mutancy. And what does it do? It just basically partitions the two nucleoids from each other so that they can form two cells, okay? But what's interesting about them is that they can form, they can form other things. They can form filaments, they can form, these rings like we talked about, and other 3D structures. So it's very similar to the way that the protein tubulin in our eukaryotic cell can form our microtubules or centrioles and other cytoskeleton components. So we use the term homology. So homology basically means that similar structures with the same purpose or similar purposes. So comes from basically the same origin. So we actually think that these FtsZ proteins are basically the ancestors of our tubulin that makes up our, the protein tubulin in our eukaryotic cells. So what does tubulin do in our cells? Well, our mitotic spindle fibers are composed of microtubules that are basically polymers of the protein tubulin. So this FtsZ protein is very active in prokaryotic cell division, and it's very similar to tubulin in the structures that that are formed in our eukaryotic cell division. So it's possible that this FtsZ in binary fission and our mitotic spindle in our mitosis, that they are related. Okay, so that there's some ancestry involved there. All right. So so there's some similarities there that we believe that perhaps these are precursors to our tubulin proteins. All right. Okay, so let's do some review questions. So what eukaryotic cell cycle event is missing in binary fission? So in binary fission, do they not go through cell growth, DNA duplication, mitosis, or cytokinesis? Well, they don't go through mitosis because they don't have a nucleus to undergo nuclear division, right? So C is the correct answer. Here's another question. FtsZ proteins direct the formation of a blank that will eventually form the new cell walls of the daughter cells. Is it a contractile ring, a cell plate, cytoskeleton, or septum? So the FtsZ proteins direct the formation of a what? If you said a septum, I would agree with you. So the septum is what forms, that will eventually form the new cell walls of the daughter cell. Right, so that's it for 6.4 prokaryotic cell division. So by now you should be able to describe the very easy process of binary fission in prokaryotes and explain how FtsZ and tubulin proteins are examples of homology. So in the next video, we're going to switch gears. Instead of looking at cell division in our somatic typical body cells, we're going to look at how we can undergo basically instead of asexual reproduction with producing identical daughter cells, we're going to look at sexual reproduction. So how we end up producing individuals that are genetically unique through sexual reproduction.